This study looks at our blood at the tiniest possible level, one cell at a time. Scientists use a new approach that lets them see both the genes and the proteins inside each cell. By doing so, they built a super detailed map showing how all types of blood and immune cells are related. It's like switching from a blurry map to Google Maps with every street labeled. Some cutting edge lab tools that were used are AppSec and single cell RNA sequencing. This both reads a cell's protein surface ID badge and its gene activity at the same time. Cell hashing, it's like putting name tags on each sample so many people's cells can be tested together without getting mixed up. This assists in testing a bunch of cells without the error. Smart sequencing to and sorting, which double checks that the computer's labels match the real cells for accuracy. An NRN algorithm, which is a smart program that compares simple lab data to the fancy single cell map that comes from this study. Computational panel design, which is a computer program that automatically picks the best markers to sort and identify blood cells more precisely. Some older methods could only tell part of the story. Flow cytometry shows what proteins are on the surface, but not necessarily what's happening inside of the cell. RNA tests shows what genes are active, but not how the cells look or behave. This new method does both of them at once. It links what a cell looks like to what it's doing, essentially creating a full profile of the entire cell, what it's doing and what it's looked like, which makes it more accurate in how we can treat diseases. And because of the computer tools, even normal hospital lab data can now be compared to this super detailed map. Some advantages over the conventional methods. This approach is better because it gives a clear picture of each cell and not just an average. It's way more accurate because it can find rare cell types that older tests missed. It's much cheaper and faster because computers choose the best tests instead of people guessing which tests may be necessary and which are not. So instead of having to run a full gamut of tests, now with this computer technology, it can specifically say we need either one or two tests or another test, which saves a lot of money and time. It's also easier to use in hospitals. The data can plug right into everyday lab machines. In short, it's like upgrading from a black and white photo to a full color 3D image of how our blood really works. In the real world, a similar technique called site sequencing was actually used in another study on a rare type of leukemia called mixed phenotype acute leukemia. Researchers looked at each cancer cell proteins and genes. They discovered a special stem-like group of cells that seemed to drive the disease and created a test that predicts how well patients might do. This shows how these new single-cell tools can move from research labs to real medical use, helping doctors better understand and treat blood cancers. By studying the blood one cell at a time and also our immune cells one cell at a time, seeing both its outer identity and inner workings, scientists are transforming how we diagnose, track, and treat diseases. If you would like to read any of the studies, I have put them in the description below. Thank you.